Okay.
All right, then.
So we should stop chatting and I should make my hair. Huh?
All right, thank you, everyone. Thank you for joining. Thank you all for staying with us till now. This is going to be the last session for today. And we have a very important guest in the room, which is Despina Moschou. And she's a lecturer, assistant professor in electronics and electrical engineering, Center of Biosensors and Bioelectronics and Biodevices in the University of Bath. And we're happy to have you here as one of our speakers from the Greece section. And I will also still have Iris with us in the room. So I will let Despina do the little introduction and let's get to it. So Despina, the floor is yours.
For inviting me and giving me the opportunity to speak in such an interesting audience. It's always nice to speak with young engineers and talk to you about something that I'm pretty sure you don't really get to hear a lot about in electrical engineering. So can you see my screen? Do I just carry on?
Yes, we can see you very well, Despina.
Fantastic. So first of all, I will explain to you a little bit who I am and how much of an electronic engineer I am, what I do in the University of Bath, and in particular in our research center in Biosensors, Bioelectronics, and Biodevices, and then I will get right down to science. So explain what LabonChip is, the particular niche that I'm actually leading in labs on printed circuit boards. I will get into the example of COVID because it's topical and I'm pretty sure you all want to know what electronic engineers can do in this arena. And then I will explain more about things that we have been doing before COVID and we will keep doing after COVID because the world did not finish with this virus. So first of all, I always like to have this slide on, especially when I talk to electronic engineers, to explain to you that I am an electronic engineer. And the fact that I'm doing biomedical engineering or these LabonChip applications and research that I'm doing is on top of being an electronic engineer. So anyone could do it. My basic degree is in electronic and computer engineering with the specialization of microelectronic circuit design. I had nothing to do with medicine, biology, chemistry, absolutely nothing. My PhD was also pure microelectronics. It was done in NCSR Demokritos in Greece. On TFT fabrication, so thin film transistor fabrication in collaboration with Sub Labs of America. Later on in my postdoctoral research, it is when I started going into LabonChip and started gaining all the cross-disciplinary skills that at the moment I have. So first working in Demokritos, also in Athens, and then relocating to the UK, first to the University of Southampton, and later on in starting my own independent group in the University of Bath, which I will elaborate more. I also like showing the pictures that you see there, both of my professional career, so clean room fabrication, which is something that I've done for several years, but also on top of having caring responsibilities and a family, and on top of the nice picture you see here on the right, is basically my university friends. And a lot of them had to do with IEEE. So I just wanted to make the point of how important it is to get involved as a young professional in this kind of institutions on any aspect. Now, what I'm doing now. So since 2016, I started my own research group in the University of Bath. Bath is called Bath because it is actually named after baths, Roman baths, the ones that you see here on the right. It's a Roman city, one of the oldest in the UK, and it is the only natural bath that you can find within the UK. It's a very pretty city. It is actually a UNESCO heritage city, so it is protected. Its architecture is protected. Very close to London, over here in the west of the UK. Our university, you can see here pictures of our campus. We are 50 years old, so we are a very new university, which makes us also very dynamic and very rapidly expanding. So my research center, the Center for Biosensors, Bioelectronics, and Biodevices, is only 2 years old. and very, very quickly expanding in the areas of biosensors, bioelectronics, and biodevices. In this specific research area, we are actually the largest research center within the UK. Our mission is to develop technology that improves biomedical diagnosis, environmental monitoring, and also scientific understanding of basic biological functions, but we are very focused on real-life impact. So we are not really oriented towards academic impact. This also happens, but we are driven by the end users in order to see our devices used at the field. And of course, in order to do that, our research center is highly cross-disciplinary. It involves all the related engineering disciplines, electrical, mechanical, chemical engineering, basic scientists from chemistry, biology, biochemistry, and of course medicine, a lot of hospitals, a lot of end users from various areas. And we are very rapidly expanding our international network of collaborators. We have, before COVID, we used to travel a lot, all of us, to all of the different parts of the globe. So we have collaborations now ongoing with the US, with Singapore, with India, with South Africa, and we just keep growing, basically. We like going international. So let's get down to science, and I will explain to you what my research area is, which is LabonChip. Literally, it is what you see on the picture on the left. So what we are trying to do is shrink down the complete biochemical laboratory onto few centimeter microchips. This is not unprecedented, of course. You are all electronic engineers, and you know that this is what happened with integrated circuits. Computers, back in the very, very old days, used to take up complete rooms, floors of hardware, until we invented and shrunk down everything to a small microchip, integrated circuit. How did we do that? We basically had integrated solid-state devices, shrinking down all the components of a circuit. So the transistors, the diodes, the capacitors, the inductors, and all of the different electronic components we needed. In LabonChip, it's not that easy, because most of the components that we need and the reagents and everything we need to handle is liquid. So solid-state devices will just not cut it. We need to deliver liquid samples on a small microchip. This, by its nature, involves microfluidics. So channels that deliver, microchannels that deliver the reagents and the samples where exactly we want them on the miniaturized microchip, valves and pumps that can control very accurately where the liquids are dispensed and when, and of course, detectors, biosensors that quantify the analyte, the chemical, the molecule that we want to detect. And of course, you can get much more complicated functionalities at the liquid arena by separating different components of the liquid sample that you have, or mixing different chemicals together so that you can create a reaction, and there is an endless list of more components. I used to have this slide to explain to people why this is important. Now COVID made it self-explanatory. I mean, the advantage of having a MiniLab in your hand is something that could make pandemics managed much more differently than we are handling them now. There is a very limited capacity in biochemical laboratories. We cannot just have one biochemical laboratory per person in the world, unless we have a MiniLab, and we give everyone a MiniLab. This is exactly what happened with computers. Before, when we had only mainframe computers, very few people could access information. As soon as we shrunk down information and people could have computers, laptops, smartphones, information just became available to everyone. So this is the unique advantage that LabonChip technology can give to society. An indicative application of how big a difference and an impact could make in real life, of course, is point-of-care medical diagnostics, which is what I will focus today, but it's not the only area. So we do work, we used to, and we still work a lot with environmental monitoring. There are a lot of defense and security applications, a lot of applications on food safety, regenerative medicine, so organ-on-chip devices that also have a lot of attention recently, chemical synthesis, and who knows what else. Again, before COVID, there was a lot of interest and a lot of investment from a lot of companies, multinational, a lot of startups emerging with new technologies. The interesting part, what I always wanted to point out here, is that the main stakeholders are not only pharmaceutical companies. It's actually 50% pharmaceutical companies and 50% technology companies. You would see Sony, you would see General Electric, all the big names in technology, they saw this niche. And because this field is cross-disciplinary, everyone belongs to it. So everyone can have stake in it. And now it has expanded, and basically anyone who has any interest in diagnostics just wants a piece of it. Now, I talked to you before about some of the components of LabonChip devices. Indicatively here, you can see some pictures of some microfluidic channels, very complicated in different materials, microvalves and micropumps. I'm showing here indicatively some pictures of just a version of a technology for these kind of applications. If you Google each of these terminology, you can find thousands of papers of different ways of implementing them. The same with separators and micromixers, which are the basic components of a LabonChip device. And of course, in terms of sensing, chemical and biological sensing, this is a whole new field by itself that is focusing on quantifying somehow a biochemical reaction, a biochemical component. So I showed you all of the components. How do we now build a chip from them? This is the integrated circuit, because we put everything together in one single platform. How do we put these components together now? There are different technologies. The one that started first, because we were all familiar, like myself, we came from the field of microelectronic fabrication. We were familiar with silicon processing. So what we did was basically use what we knew, clean room fabrication on silicon substrates, to fabricate integrated LabonChip devices. And this is the first technology that you see here on the left, CMOS. It has the advantage of having a very good global infrastructure for manufacturing commercially such devices. However, it has the problem when we talk about LabonChip devices that the overall cost when you integrate microfluidics on them is not cheap anymore. Silicon microchips became cheap because we could fit a lot on a silicon wafer. And we can fit a lot because the microchips are a few millimeters in dimension. When you want to build centimeter scale devices, you cannot fit thousands of them. You can probably fit maybe 10 or 20 of them on a silicon wafer. This by itself means that the devices in the end will not be cheap. And this is a problem. The same problem is with glass technology. So glass is also one of the materials that was used in the beginning for LabonChip devices because it's very well known, it's transparent, it's biocompatible. However, it is a very expensive material. So we can have laboratory prototypes, but we cannot have devices that everyone just uses. This is why plastics, all kinds of polymers came into play. They are very cheap. There is a very big variety of different plastics with different properties. So we could make LabonChip devices doing literally depending on the properties that we cared about. However, when we want to integrate electronics and make smart devices, because electronics is what makes any device smart, we can't put them on polymers in a straightforward, upscalable way. We can do it in the lab. We can make a prototype, but then when they order, somebody is interested and we want to make 1,000, 1 million of these devices, it is virtually impossible because there is no such factory. So there is a big bottleneck there. Paper gained a lot of interest recently, again, because it's a very cheap material. You can print things on paper. A huge advantage because it's extremely simple. However, it has the problem that you cannot make it smart. You cannot put electronics on it very, very easily. So it's still under discussion. So I gave you a lot of processes and no solution. Everyone is problematic. of course, apart from the one that I'm pioneering, because I have to blow my trumpet a little bit, but you will see why I believe in it. So basically, this is where Labon printed circuit board devices come into play. And as a pure electronic engineer, I would focus on printed circuit boards. Why? Despite the fact that they sound low tech, they have actually been sidelined for no reason. So basically, I'm not the first person to say that we should be building MiniLabs and printed circuit boards. It was first suggested in the 90s by the people who first conceptualized LabonChip technology. However, at that point, people were focusing on proving the components, the microfluidic components. Nobody was really focusing on the integrated thing because we were very far from it at that point. So at that point, especially polymer processing became a prevalent technique. But right now, after 20, 30 years of research where we have proven all of the components and people are focusing again on how do we put now everything together on one microchip, PCBs seem to be the ideal integration platform. Why? First of all, there is no problem of building new factories. The factories are already there. There is an infrastructure globally. It is low cost. You know how cheap printed circuit boards can be or how expensive, depending on what you want to manufacture. In terms of microfabrication, they are 100% compatible with LabonChip devices. They are not nanometer scale features that we want to make. They are actually micrometer scale, and they're 100% compatible with our dimensions. And of course, the problem of integrating electronics is solved because they are made for electronics. So they are an ideal platform. But okay, we know that printed circuit board technology exists, but can we make LabonChip devices on them? I started making the first ones myself when I was in Athens, started working on it in 2010 in NCSR Demokritos. And what you see here on the left is basically handmade at that point. DNA amplification device on a flexible printed circuit board. So basically the cost of this device is minimal. I would say pennies, because it's a very, very simple technique to use. You basically just fabricate resistive heaters on the bottom side, and on top you make with simple photolithography of dry film photoresists microfluidic channels. And what you fabricate here within a few centimeters, I think this was 6 by 8 centimeters, is basically a mini thermocycler. What is a thermocycler? So in any kind of genetic analysis, and we use genetic analysis a lot in biomedical diagnostics, when we want to identify a pathogen or identify in paternity tests or in forensic tests, when we want to identify any living organism, we need a thermocycler so that we can amplify the genetic information, the DNA piece that we get from the living organism. Usually within a cell, there is very little amount of DNA, so we need to amplify it. The process of amplifying it, basically the most widespread process of amplifying DNA is called PCR, polymerase chain reaction, and is done normally in mainstream labs in thermocyclers, so big pieces of equipment that do thermocycling. This is a mini thermocycler. So we did amplify DNA, as you see. A regular thermocycler takes about an hour to do the amplification of DNA. In our case, we went down to a few minutes, 5 to 10 minutes to do the full amplification. And then we proceeded with integrating it in a bigger PCB, integrating it with the sensors. So amplifying the DNA, but also quantifying it in a silicon microchip. That could produce electrical signal proportional to the amount of DNA that we detected. This worked. This is handmade by myself. But apart from the fact that it worked, what I quickly realized is that this is not upscalable. And the reason for that is that you are integrating two different materials. You have a PCB and a silicon microchip. This electrically can be done. Fluidically, you will always have leakages. There will be very few devices, and they are all handmade, but they will always have leakages because interfacing two different materials is not something you can easily do when you're talking about fluids. Even a small leakage can actually destroy the complete device. So I started looking into ways of putting the biosensors on the PCB itself, not having to integrate different materials. And I found in literature that many people have been doing it. They have been using the roots plated with noble metals, because for many reasons we want noble metal coatings so that no reactions, unwanted reactions happen on the surfaces. On this device, on these PCB roots, we can immobilize biomolecules, and we can have reactions happening on the roots themselves. So when I moved to Southampton, this is what I tried to do. So for a different application, in this case, we wanted to quantify proteins for tuberculosis diagnosis. I wanted to see if I can build an electrochemical biosensor on the printed circuit board itself. And taking it even a step further with the team that I was working there at that point, I wanted to do it on a commercial device. So not a device that I made by myself in the lab, but something that a conventional PCB manufacturer could make. And we did succeed. So as you will see here, this is my first design of this ELISA test. It was a few centimeters again in size, and could do electronic quantification of interferon gamma, a protein biomarker that is indicative of tuberculosis. Here you can see how well it performed. The final device, again, as I told you, commercially fabricated, which I think it was a first. And it is not as easy as one would imagine, because PCB manufacturers are not familiar with biochemistry or with LabonChip. They know how to make electronic circuits, so it required a lot of skill to communicate and educate manufacturers on how they could do and explain to them what exactly it is that we want to do. But we did it. And what you can see here is basically that we managed to quantify our protein in a way, in a fashion comparable to the standard fluorescence-based technique that they use in the lab, at that point with our collaborators in Imperial College Hospitals, Charing Cross Hospital. But as you can see here, I was not satisfied again, because the error bar here is 3 times larger than the one of the conventional technique. And I knew why this was happening. And I thought this was because of the gold plating that was unreliable. I could see it peeling off. So I could see that this is one of the major issues that this technology had. So later on, I moved to Bath and I started my own group. And I tried to make this, I was certain that this is the way forward. I wanted to expand more and solve all these problems that I showed you, and a lot of more that came up. So we started working first on solving the problem of this plating and minimizing the error bars, making the sensors more reliable. And we did find out that, as you will see here, we tried different platings, commercial platings of PCBs, and we did find that they play an important role in the performance of the devices. So if you buy any board from any manufacturer, do not expect it to work in the best possible way. There is some development work to be done there. We have been working with a lot of them to optimize their processes for biochemical applications. So, and we are hoping that in the next few years, more factories will be able to deliver what we want. But we did demonstrate here in terms of DNA sensing that we can have it on a commercial board, the one that you see over here. And we reached the lowest limit of detection in DNA quantification that has been achieved so far in such devices. We demonstrated very clearly the importance of the surface characteristics of the printed circuit board. And we also tried to integrate the DNA quantification biosensors that I showed you before with the DNA amplification that I showed you earlier, and we did, again, as you see here, in a commercial device. In parallel, my group has been expanding a lot on different applications, not only about DNA. So my PhD student that you see over here, Ruros, has been working for the past 3 years on a device for sepsis. Sepsis is a major problem. There is no diagnostic, actually. I mean, there is not a quantified test that the doctor can do and definitively identify that you are going into sepsis. So what we did in our project was develop a small microchip, again, a Labon PCB, that can quantify protein biomarkers that are indicative of sepsis, telling the doctors very quickly that this person is highly likely to get into sepsis within the next few hours. This, in collaboration with the Wyss Institute in Harvard, we actually demonstrated that we can quantify simultaneously 3 analytes, and we are actually right now designing and testing an integrated device, which I'm not ready to share with you yet. So while doing all of those things, most of my students and most of my group, for some reason, are very commercially oriented. I think it's this whole startup craze that has been going around the world, but I understand that they see the commercial interest in what we do, so they did start a company. I'm also a co-founder in the company, but I'm not involved in, I don't mess with their business because I'm not the right person to be doing business. I only support academically and scientifically what I can. And they did start selling these PCB sensing devices and in the first instance, to research groups. But for me, the most interesting thing is not that my research group did it, it's also that there was competition from people who are not us, which means that maybe we're onto something. So we were very happy that this happened. And then COVID happened. And all of a sudden, we were busier than we were before. So because I'm talking to an electronic engineering audience, I will say a few things, first of all, about what this virus is and how it is diagnosed. And then I will explain to you how we as electronic engineers have been working on it to make, hopefully, our lives better in the next few months. The name of the actual virus is SARS-CoV-2. COVID-19 is the disease that is caused by this virus. The name is SARS-CoV-2 because it is a virus that causes severe acute respiratory syndrome. It's a coronavirus, hence CoV. It's not the only coronavirus, there are a lot of them. It's not the only virus that causes severe acute respiratory syndrome. There have been others, there was SARS-1. And this is number 2, because it is the number 2 that we have recorded. It's a new coronavirus that causes SARS, but it's not the first one. Okay, so like the other coronaviruses, it is a very small particle, like the one you see here on the left, and a very simple structure. Viruses are not living organisms. They are not considered a microbe for this reason, because they are not like bacteria that are actually alive. Viruses are extremely simple. They just have a protein shell around them and genetic material inside them. These protein spikes that you see here, basically they attach to specific cells in our organism, in our body. In this case, they attach to the back of the throat, and they open up their envelope and start releasing the genetic material and start multiplying. So basically, when you want to detect a virus, you want to isolate them from wherever they have attached. If they don't attach, you don't get sick. They just get thrown out of your system. You only get sick if they attach, because they start opening and releasing their genetic material. So if you want to detect them, you need to catch them first from where they attach, which is why you have seen all the swabs that go in the back of your throat, trying to catch the virus where it has a highly, it is more likely that you will find them. And then you need to break them apart and identify their DNA and see if it is indeed this virus. This is the molecular test, if you have heard in the news. Apart from the molecular test, it's currently considered the most accurate test. No test is 100% for many reasons that I will explain to you now, but it is the most reliable test that we have at the moment. There is also the possibility of not detecting the virus itself, but detecting the outcome of the virus. So if you become infected, your body responds. And what it does is basically increase the levels of some other proteins in order to fight back. There is the possibility of quantifying these proteins, so antibodies or antigens that are produced within your blood. This is serological testing, which is done with a process called ELISA, similar to the microchips that I showed you earlier. So the process for molecular testing is, first of all, extracting a swab from a patient, so trying to catch viruses, because they do not exist all the time, and they are not that easy, especially if there are very few of them. You never know if you're going to catch them, but let's hope that you find them there and you catch them. And then as soon as the swab goes into the central lab, we need to have what we call virus elution. So we need to release the viruses from the swab. Then we need to extract the RNA, the genetic material that they have inside. And then we need to amplify it with a DNA PCR polymerase chain reaction, the process that I explained to you earlier, and find out if the sequence of the virus is there or not. In terms of antibody and antigen testing, what we do is the ELISA process that I referred to earlier. So this is also a very laborious laboratory process, which involves these 96 well plates that you see over here. There are a lot of assay steps, so a lot of pipetting, like you see here in the lab. It takes hours, probably 2 or 3 hours, to do all of the steps and the incubations and all the wait time that you have to do. In the end, you end up with a 96 well plate like the one that I show you here, which shows in colorimetry, so by looking at the color, whether the antigen or the antibody is there or not. So you insert it in a benchtop piece of equipment, the plate analyzer that you see on the right, and you get the result of whether there is presence or absence of the antibody. These are processes that take hours, even in the best of the systems. What can we do? So you heard the World Health Organization saying that we should be testing every suspected case. Is this really feasible? With the current centralized laboratory facilities of any country, it is impossible. We cannot test every person in the world at the same time in one central laboratory. We just don't have the capacity or the reagents or anything. However, this is needed, as you all know. The only way to do that is with MiniLabs. If we are able to distribute the workload and of testing in different at community level or at personal level. So we are working in my group, but also I need to tell you that there is so much activity in this area, as you would imagine, both from academic institutions all over the world, from companies all over the world. Everyone is pushing for this, both to help and also for personal interest. But there is a huge activity, so I will focus here only on the stuff that we do in my group. So we do work on the molecular diagnostic platforms that we have developed from the previous years in collaboration with local hospitals in Bath and also in Cambridge with our partners at the University of Glasgow that have a faster DNA amplification assay. We work with different countries, so we have an ongoing collaboration with the Ministry of Health in Uganda to test our devices there. And there has been a lot of activity with policymakers, with the diagnostic industry partners. Everyone is interested to do something and sell something for COVID. We are not allowed to say everything at this stage. Maybe in the next few months, when things are solidified, maybe we can start disclosing more things. But at the moment, there is a lot of activity and a lot of interest. On the molecular diagnostic, which is a substitute for PCR that you see now, our partner hospitals have told us that actually they have a big problem, not only on diagnosing, but also on prognosis of whether COVID patients need to go into the intensive care unit or if they could just go home. In order to see that, you don't need to identify if there is a virus, but you need to quantify the levels of some inflammation markers that they have in their blood. At the moment, there is no fast way to do this. So they need to order ELISA laboratory tests. So our sepsis device is actually being tested now and in the next few months to see if we can help with triaging COVID patients. These are all under development. I don't have results to tell you how efficient they are or how fast they are. It is work that is just being carried on at this stage. And of course, on another level, diagnostic level, we have the immunity testing. So after you contract SARS-CoV-2, you might get sick or you might not get sick, but in either case, you have antibodies in you. This means if you have antibodies that you are immune. I'm not qualified to say for how long or any of these big questions, but it is important to know that you have some antibodies in there. We can do that with pregnancy type tests, what we call in the industry lateral flow assays, like the one that you see here on the right. What my company recently received funding to do is to make these tests smart. So basically, as you see them here, they're like pregnancy tests. So you need to look at them and see if a person has antibodies or not, whether there's a line or forming or not. What Biotip is doing is actually making electronics to be integrated with any of these lateral flow assays that are being developed by multiple companies around the world, all waiting to get regulatory approval from the local policymakers. And by having them electronic, first of all, they are much more accurate, so they can detect much lower levels of antibodies. And you can transmit the information immediately, electronically, and safely to a remote monitoring body that would be interested to see how many people are immune and where and how. So we do believe that this is also a very interesting application for our technology. Now, you've seen how important, I think, that it is quite important to build such devices for this pandemic. However, the world will not finish with this pandemic. There will still be more disease. There will still be more outbreaks. There will still be more viruses that will come in the future. So our research is not limited there. That's why the title of my talk today is around the example of SARS-CoV-2, which made our case for us on why this is important. But our research needs to go beyond that. And it is. So what we are doing in my group is basically building a framework for LabonChip developers so that the next pandemic, we are ready. So we don't have to do the research that we are doing here now. We are ready to design devices that can address the needs of the next outbreak. So we are basically trying to replicate the electronics process using software with standard components in them that we can start picking and placing to meet the clinical requirements that we need. We are working with a lot of manufacturers, educating them and helping them provide this service to LabonChip designers. And we are working a lot also with engineers, educating them on how to do this, how to become LabonChip developers. There is a lot of work to be done. It cannot be done immediately, but we have progressed a lot. You will see a lot of our work also on YouTube if you're interested. And there is also a textbook coming. There are many things coming in the next few years. We have worked a lot with wearable devices. In this case, you will see an example of a painless, reliable, disposable patch for glucose monitoring for diabetic children. In this case, we were working with partners in Turkey because actually diabetes is a huge problem in low and middle income economies. And unfortunately, there is no painless diagnostic at the moment. This is what we focused on. We focused on doing this in a cheap way. So again, we are using a Labon PCB approach, but in this case, a flexible device because we need to have a patch. We have integrated microfluidics to extract fluid from microneedles that are integrated on our device, and again, seamlessly integrated electrochemical glucose biosensors to quantify the level of glucose in the interstitial fluid that we are extracting. I will not go into too much science because this might be too much for you. You can find all the information in the journal papers that I'm citing, but we have managed to quantify it very accurately in very low concentrations. And here you can see a device, a very rough prototype that we did in the lab, proving that we can have the concept that I described to you earlier. This work is actually right now being expanded further through my industrial fellowship, the Royal Academy of Engineering funded fellowship with a company called Kaura Ltd. who are developing also such kind of patches, mainly for sports training applications. We are also trying to print electronics, so on top of the PCB devices that we have, print semiconducting devices to see if we can push further the performance of our devices. We recently showed that we can have electrolyte-gated field effect devices printed by simple desktop inkjet printers. Working with our collaborators in India, we managed to quantify bacteria, whole life bacteria in simple water samples. Again, ongoing work and ongoing collaboration. We have been working with water quality parameters. So in this case, we are making an integrated labon PCB device for quantifying water conductivity, the pH of water, but also whole total bacterial load. So identifying whether water is contaminated or not on a simple, again, microchip device. And here my student also got a bursary to work on her final year project further. Managed to get also the electronics to make it smart and also make an app to go with it. Finally, we're working, to my big surprise, with Airbus. So basically plane manufacturer. The first time they approached me, I asked them if they really know what they do, because I don't do airplanes. Nonetheless, they were interested even themselves in biological applications. I am not again allowed to say exactly what their interest is. You will hear more in the next few years probably, but we started working on the concept of quantifying water in oil samples for applications of their interest. Stay with me, and I think in a few years you will hear more about it. This has become again very topical with COVID. You all know the troubles that the airplane industry and the air traffic is now at the moment. I thought this would reduce their interest in what we do. It actually increased it further, and we are expanding a lot, potentially also on diagnostic applications. But again, I cannot say many things. I talked to you before about organ-on-chip devices. We can make microchips to simulate the operation of different organs of the human body. The reason to do that is A, for understanding, because you will be impressed by how little us scientists know about how our organs work. So we do need small simulators of little cells so that we can perform and understand their operation, but also to test drugs, because at the moment most of drug testing is happening in poor little rats, mice, and rabbits. By using such devices, organ-on-chip devices, we could reduce this to a minimum. So indicatively here, you can see a brain-on-chip device that we have done in my lab in collaboration with Dr. Rocha. Finally, an indicative application, I wanted to let you know that what we have tried is producing electricity on the printed circuit board by glucose-containing samples. So you can actually produce electricity for electronics without using a battery, without plugging anything anywhere, just by spitting and just by using sugary drinks. You can actually do that. We recently published the work. It's not finished yet. It's still very low power, but my friend and colleague from chemical engineering is working on making it more efficient. As I told you, it is very important for what we do to engage with the industry, make them embrace, help them embrace the concept so that they can open up to a new market and learn new skills. It has gone very well. We have a very strong link now with the European PCB industry. I've recently been doing a lot of workshops for them and with them, writing a lot of articles. There is a lot of engagement, and I'm hoping to an avalanche effect and very quickly expand even further. So I think I'm within my time. So I would like to wrap up. I told you a lot of information. I hope it was interesting for you. So concluding, I just want to sum up saying that infectious disease have always happened. They were there. They didn't appear now. They have been there as long as humans have been here, and they will carry on being around. This is why we have been working on these technologies before COVID, and we will carry on working for them after COVID. There will be a world after that. The important thing for me is what this pandemic has shown is the definitive proof that we need high technology solutions. We are actually fighting this pandemic the same way we fought plague, the previous pandemics, when we did not have technology, which I think is wrong, plain and simple wrong. And it's the job of engineers to help and create new ways of handling in a smarter way this kind of challenges without having to lock people up. Anyway, there is a way to do it. I've showed you a lot of examples of technology that is out there. I've showed you that it is possible to have cost-effective manufacturing of integrated LabonChip devices. And I've talked a lot about the specific research that I do on labs on printed circuit boards, which I pursue because I believe in it. I think it's the way forward, because there are so many materials and processes commercially available that I don't know of any other way or any other technology to use. We have proven prototypes demonstrated for a very broad variety of applications now, which shows that it's possible and commercially fabricated. The dimensions and the requirements for LabonChip technology are 100% compatible with what can be achieved, and the factories are there. So we don't need to worry about mass production. We don't need to make new factories. I think this is a huge advantage for any technology, simplifying the translational routes. So with that, I would like to thank everyone. This is not everyone. This is just a few people from my group and a few collaborators. I told you we work with the whole world. This is not, again, inclusive of everyone. It has been a bit difficult not to be able to travel and talk to all of my friends everywhere and make new friends. But this is a job, research is a job that is international. And this is a huge advantage for technology to move forward. And this is why I like initiatives like yours that bring together people rather than pull them apart. So with that, I would like to thank you, and I'm very happy to answer any questions you may have.
Well, thank you very much, Despina. That was wonderful. I think most of our audience were able to enjoy this, and they stayed with us to the end. And I think we have some questions. For those people that have just joined us, if you have questions, please feel free to ask in the Ask a Question box below. But I think I'll just go quickly to that box so that I'll be able to read them out for you. And at the same time, you feel free to attend them, right? Okay. So let me read out the questions so that you'll be able to answer them. We have the first question coming up that says, we know from Theranos that having something so tiny imposes many limits. She further asks that, where do you think we will be in let's say 10 years from now regarding labs and on chips? So if you can just provide that response.
Sure. I'm very happy you are familiar with the Theranos story. I always keep it on my slides for people who are not, to look at it and understand what happened. It was not a problem of the technology, it was a problem of the founder. So we should not be blaming the technology for the fact that people do not use it properly or do not understand what they can do with it. It is true, as you said, that for many applications, a tiny drop just does not contain enough molecules. It's impossible for certain applications, not for all of them. So if you are looking for one bacterium circulating within your whole system, it is impossible to find it in a drop. I agree with you. So this is a limitation, a major limitation, which is why when we design LabonChip devices, we consider from the beginning what is the sample volume that needs to be analyzed in order for this system to have clinical value. Or for water, sometimes we are looking for one bacterium in a liter of water. So one of the major challenges for us when we design LabonChip devices is how to isolate this one bacterium from 1 liter of water. These are important questions. This is the objective of the research that we do. There are strategies to address all of these issues. The problem with Theranos was not that it was not possible, it was the fact that they were hiding them and they were not trying to solve them, which is not the way to go. So I do believe, because I do know the field very well, and I know a lot of people who are doing this and developing devices for applications that make sense, I believe that within 10 years from now, and especially in light of the pandemic that we all live, I'm pretty sure, I don't want to give a percentage, that you will see them in real life. Maybe not in 10 years, maybe in much shorter time than this.
Okay, thank you very much. The second question is, what are the biggest limitations nowadays?
For limitations of the technology in general, on COVID, if you want to be a little bit more specific?
I think the technology, she didn't actually specify, but I think it's on the technology.
I think the main limitations are not technical. They are about proving in real life, providing one success story that has been widespreadly used by everyone, like the glucose testing. If we can show, it's mostly commercial, as you understand, rather than technical. If we can show this, then on a technical level, we can do a lot of things. On a technical level, in my opinion, the biggest limitation is integration, seamless integration. Not so making multi-component LabonChip systems, 2-component, 3-component, 4 components, including sample preparation.
Thank you very much, Despina. I think that was well taken. The next question is, how is it that the fluid do not interfere with electronics functionality on PCBs?
They are actually multi-layer devices, so multi-layer PCBs. So basically the fluids are on a different level than the electronics. You try to keep them apart. And this is also part of the work that we are doing in my group, is making design rules so that we can explain to people how they can, when they design them, they should keep them separately. But the key here is the multi-layer capability that we have with PCBs, that we can keep the fluids on one side and the electronics on the other side.
Okay, thank you. Another question is from Lucy Patricia, and she asks that what are the potential challenges of COVID-19 test kit diagnostics reliability? Can mobile phones be employed as healthcare test tools?
Starting from the mobile phones, yes, I think the fact that we have such a powerful computer in our hand, I think this is huge, and they should be employed. How? What is the best way to use them? That is a question. It's not answered yet. But that's why most of us, when we develop tests of any kind or diagnostics of any kind, we try to have them integrated with a tablet or with a mobile phone, because everyone has them. So it is a great resource, basically, that we can use. Challenges on reliability, a lot of them. Basically, when you do anything biochemical, there is, first of all, the problem of the stability of the biochemical reagents, which is a problem addressed by biochemists. It's not something that engineers can do. But we also have problems of repeatability, reliability, which are to be addressed by ourselves, making sure that our devices are the same. This is why I put a lot of emphasis always on commercial fabrication, because commercial fabrication means reliability and repeatability, knowing how many devices will work and how many will not work. Whilst when I do something in my lab, I cannot guarantee that. I don't have fabrication standards, I don't have ISO processes. So that's why I think it is very, very critical to have the collaboration of the industry in this. But I also want to make a point here that even if we make all of our devices 100% reliable, there is always the problem of the person who collects the sample. if the person who puts the swab in a sick person does not do it correctly, even if your device is 100% reliable, the test will fail. So if we could somehow, and there are a lot of groups working on different ways of sampling that are simpler, maybe analysis in breath or in spit, because any methodology that takes away human handling will increase reliability a lot.
Okay, thank you very much, Despina. I think we have the final question, and it's like, which advice will you give to a circuit designer with background in telecom electronics and zero background in biofield that would like to change or start in bioelectronics?
My advice would be to do a PhD. This is the best way to learn, really learn and understand. I think I would advise a PhD. If not possible to do a PhD, a master's degree. And within your master's degree, try to get for your thesis, for your final project, do a research-focused project. This is how you can learn these things, because this is a field that is rapidly expanding. Everything is happening now at the research area. So the closer you become, so we don't have textbooks yet on these things. Everything is on journal papers. So if you don't have the education either at master's level or at PhD level, even if you try to read journal papers by yourself, I highly doubt if you will understand a lot of things. I think you need an academic with you to explain to you things. So I would suggest education either at master's or ideally at PhD level.
Thank you very much, Despina. It was nice having you. And before we go, I would like to invite Aria, which is a young professional ambassador from Greece section. I would like to come on the air to join us on stage to give a vote of thanks to our guest speaker here. And also, I will also want to say wonderful outing for the Greece section. You guys did wonderfully bringing us Despina, and our lecture was very, very well taken, insightful, and I begin to find interest in the biotech. So, Aria, can you please give a vote of thanks to Despina?
Thank you very much, Despina, for the very interesting and informative presentation. I would also like to, as I mentioned in my presentation before, I would like to thank the organizing committee, the amazing organizing committee, and of course the IEEE section board, and especially our chair, Professor Konstantina Nikita. And of course, all of you guys that you were here with us today.
Right, that's wonderful. So thank you for staying with us to the last second in the hour. We do appreciate you. We have a lot of lineup programs for you tomorrow. So please find time to join, connect with the virtual seat, keep networking with everyone. And thank you very much, Aria and Despina. We do appreciate your time, and we hope we'll be able to meet face-to-face after post-COVID. Thank you very much, and have a nice evening, and catch you all tomorrow.
Thank you very much.
Bye. Bye.